Good afternoon, everyone, and esteemed guests, distinguished colleagues. It is with great pleasure that I introduce you to our keynote speaker for the PSS Spring Seminar 2024, Dr. Benildo de las Reyes. Dr. Reyes is the Bayer CropScience Reagent Endowed Chair in the Plant Genomics and serves as the Associate Department Chair for Research and Graduate Program at Texas Tech. His remarkable journey in academia has been marked by relentless pursuit of knowledge and innovation. Prior to coming to Texas Tech, Dr. Reyes was a professor of molecular genetics and associate director of University of Men's College of Biology and Ecology. He has changed the landscape of Texas Tech, helping successfully craft the governor's research initiative that brought the Institute of Genomics for crop abiotic stress tolerance to Texas Tech University. Despite these accomplishments, everyone who knows Dr. Reyes realizes that his true passion are advancing the frontiers of plant science and guiding the next generation of outstanding scientists. Please help us welcome Dr. Reyes, a visionary leader, an inspiring educator, and a true trailblazer in the field of plant genomics.
Thank you, Rupak, and for the graduate student in the department for hosting this. And there are two reasons why I actually volunteered to do this today. One reason is because I was really very eager. I was just discussing it with Glenn a number of times and Scott. I was very eager to get this restarted again. For new graduate students who have been here only post-pandemic, we started this big time, this seminar series big time. Before the pandemic, where we started putting the graduate student organization to coordinate the whole seminar. It's part of your training, okay? So that's one of the reasons why I volunteered to be the opening talk for this seminar series. The second reason is because I have delivered this lecture. I was counting it last night. This is the 11th time that I'm delivering this lecture. The past 10 times was across 10 different countries, 3 different countries, 10 different institutions across 3 different countries, the United Arab Emirates, Japan, and the Philippines. So every time I deliver this talk, I receive a very enthusiastic reaction or feedback, both from students and scientists alike, because I believe I was able to make them think outside the box, okay? Which is my main reason in today's seminar, which is essentially to inspire you students to actually think outside the box. I have had several conversations with my colleagues in the department about the question of, are we teaching enough philosophy to our PhD or graduate students? Or are we just training them to be, highly skilled and proficient technicians that are able to execute something that is very technical, but unable to think outside the box and really put what we do, okay, all of us, what we do into a real purpose within the grand scheme of what the challenge to food security is all about, okay? And hopefully in today's lecture, I'll be able to inspire you to think outside the box, that it's not only your technical training and skills that you acquire in your PhD program that matters, but your ability to formulate the big question, okay, with regards to the bigger scheme, grander scheme of biology, okay? So today I'm gonna be putting everything that we do within the context of both philosophy and history. Okay, I'm old. I may not look too old for you, but I'm old. So I have seen the evolution of all of the technology starting from the 80s, okay, all of the technology that you are all using in today's science, okay, from the the beginning of genomics, for example, and molecular biology and its application to plant sciences. I have seen that from the initial concept of transgenic technology into more advanced iterations of what transgenic technology is all about. I have witnessed all of that. So today I'm hoping to be able to put that within the context of history and within the context of philosophy. So let me start with this one, okay? The philosophy of science, which I think what we should be teaching you guys, okay? I'm putting there a reductionism versus holism, okay? What's the Webster Dictionary's definition of reductionism? A system or process is a direct or apparent effect of its parts. A single component can explain and simplify the entire system, okay? So let's put some example in there. One causal gene for every trait, okay? That's what most of you are trying to do. You're looking for that magic gene, a master regulator that would explain a phenotype as complex as adaptation to the changes in the environment, okay? So that's reductionism. How about holism? It is defined as a system or process is the summation of all of its properties explained only by the interaction amongst the individual properties. Example, okay? The complexity of plant adaptive traits, drought, for example, okay? Adaptation to drought cannot be explained or cannot be reduced, okay, by the function of one transcription factor or one signaling gene or one signaling protein. Because you're talking about a change, a massive change in physiology that is triggered by the dynamic changes in the environment, okay? So it's a complexity that is very hard to explain if we force on looking just for one pathway or one single gene to explain the complexity of the phenotype. We're gonna go back to that in a larger context in a little while. So this is one of my most favorite slides. I always use this particular slide to make the big lesson, okay? This is, I say that this is the most important chemical reaction in the entire universe, okay? Because this is the beginning of life. This is actually fueling what life on Earth is all about, right? So a simple, well, it's not really simple. So the evolution of this chemical reaction, which we call photosynthesis, is described in this diagram from a relatively simple mechanism in primitive unicellular eukaryotes into what we know as a more complex mechanism in land plants, for example, that is responsible for all the biomass that we actually utilize on Earth, okay? And this includes food for humans, food for animals that humans use as food, fibers for protection, biomass for shelter, and even fuels and energy that, you know, fuels pretty much a lot of the technological advancement in society, okay? So let us put that in a better context, okay, by actually looking at the evolutionary purposes of plants in context of the history of food cultivation in agriculture. Why is it important? It is very important because I wanted you to understand where technology that we call agriculture this day, okay, came from, okay? Because if you do, perhaps your mind is going to be taken away from a purely reductionist perspective, okay? So let's trace the history of human civilization, okay? In the beginning, solitary lifestyle, okay? Nomadic solitary lifestyle. People were dependent upon hunting and gathering, right? Whatever is available in the biodiversity's bounty is what humans collected, okay? So let's put that within the context of plant biology, okay? During this period, plants and animals are reproducing in their native habitat. There is no tampering there, okay? Natural selection, okay? And the purpose of that is to propagate the species, right? So plants did not evolve with the purpose of feeding us. We discovered plants that as a raw material that can actually feed us, okay? And that's the beginning. Moving on to the next phase of this one, okay? A historian had proposed that humans started forming small groups because they learned how to gossip. Okay? So they learned how to gossip, and the power of gossiping is communication. So they were able to form these small groups that actually think alike. Okay. So what's the outcome of that? What's the consequence of that? They learn how to pool and share resources that they collect from nature's bounty, from what diversity could provide, okay? So again, at this particular stage, plants and animals reproduce in their natural habitat for the purpose of giving rise to the next generation. Fast forward, a lot of developments in human civilization, okay? The dawn of agriculture, okay?
I'm just going to change this.
So-
Keep talking.
Should I continue?
Yes.
Okay, so the dawn of civilization, okay, which is triggered by the invention of agriculture, right? Okay, so humans start forming more organized community, and therefore the necessity is to provide food for everybody that can no longer be sustained by hunting and gathering. So therefore, that's the beginning of agriculture because humans needed to create a technology to domesticate those diversity that's available in the wild, okay, in a managed environment so that it can produce well to sustain the needs of a more organized society. So in this case, okay, plants and animals are reared in non-native habitats, okay, and managed habitats, which we call agricultural ecosystem, okay? And this is the through the process of crop domestication, giving rise to the dawn of agriculture, and that's why we are here today, okay? Civilization started from that. Did I make the point in there? Okay, so human civilization started because of agriculture, okay? So let's fast forward to the future. I consider myself as a person who grew up during the Green Revolution era, okay? One of my mentor was the co-engineer of Green Revolution, okay? So in the modern time, okay, postmodern agricultural technology, okay, back in the 20th century, okay, middle of the 20th century, 1950s and 1960s, there was a big global issue, a big challenge to society, which is pretty much the same as what we're facing now, okay? The deterioration of natural resources, okay, climate change, okay, the disappearance of prime agricultural lands because of industrialization and urbanization, pretty much similar to the problem that was faced by society during that time. The problem then was rapid population growth and stagnant crop yield, okay? Genetics was not heavily utilized yet for agriculture, okay? So the biggest threat was the threat of famine, particularly in Asia, Latin America, and Africa, okay? The staple foods are rice, corn, and wheat, right? So those are the staple foods. So how did the political power respond to that? Let's use science. Let's use genetics, okay? So genetics, as you would guess, breeding, right? So with the power of what they call idiotype breeding, okay, there are two parallel efforts. One happened in South America and Mexico, led by Nobel Peace Prize laureate Norman Borlaug. And another parallel effort was happening in Asia, okay, led by Dr. Gurdev Kush and Dr. Henry Mitchell. Dr. Kush was my mentor, actually, okay? So these parallel efforts led to the breeding of what we now call the modern high-yielding varieties of rice, okay? That performs really well, maximize the genetic potential for yield under condition when there is ample water, ample nutrients, okay? Climate is predictable, okay? And nutrients in the soil health is pretty much ideal, right? So that was in the Green Revolution. So let's look at, in this slide, what I'm showing is that the genetic complexity, okay, of the Green Revolution paradigm is much less complex, okay? Don't worry, I discussed this with Dr. Cruz and he agreed, okay? It's not undermining the accomplishment of the first Green Revolution, okay? So, let me explain this in greater detail, okay? Rice, the modern day high-yielding varieties of rice that we are cultivating now, okay, is an outcome of the process called idiotype breeding. What's idiotype breeding? Stacking of Mendelian traits, could be morphological, developmental, anatomical, physiological, and all of those traits, okay, by Mendel's independent law of assortment, right? Stacking them together, okay? So what was being modified by this, okay? The initial prototype or idiotype of the low-yielding, low-green yielding rice, okay, has a very tall stature. I remember as a kid, we were playing in rice paddies, okay? And rice, we were running in the middle of rice paddies, and rice plants are like growing as high as like 12 feet, right? Very, very tall. So what's the advantage of that, okay? Because they're very tall, they're very lanky, okay? Because rice grows on paddy field, okay? They lodge, right? So whatever reproductive zinc that the plant produces is gonna be lost, okay? And therefore, reduction in yield, okay? They are not very responsive to fertilizer input, okay? They have an inefficient source sink dynamics. What do I mean by that? Okay, much of the photosynthese, okay, is being channeled towards vegetative growth. They're growing so tall and massive vegetative mass. Okay, And very little of the photosynthetic source is being channeled to photosynthetic sink, which are the grains, which is what we want to harvest for the crop, right? Okay, inefficient. They're photoperiod sensitive. You know, some varieties would only grow in December because of the differences in day length during the entire year, okay? Long duration, most varieties would flower after about 6 to 8 months, right? So imagine the inefficiency of that. They're very susceptible to lots of diseases and pests. So what did idiotype breeding did, okay? The mutant SD1, okay, is a gene involved in gibberellic acid signaling pathway, okay, that makes the plant semi-dwarf, okay. You would guess what's the advantage of that, because the photosynthetic source is now being partitioned towards the reproductive sink, towards the grain, okay. Short duration, farmers can grow 2 crops a year rather than 1 crop a year. So you can see here, these are all morphological traits that geneticists identified and stuck together, okay, to create these modern high-yielding varieties of rice. Let me qualify that. That is under the condition where this most important chemical reaction occurs without problem, because the reagents are there, particularly water, nutrients are there. So no problem, okay? So G by E by M, gene by environment by management interaction, okay? The green revolution paradigm would not have worked out if not for all of this, okay? Which is what is limiting to us now, okay? Abundant and clean water supply through modern irrigation technology, arable lands. Much of the world is not very much industrialized, and therefore prime agricultural lands are still available for farming, okay? So nutrients and pest control, you know, chemicals for controlling weeds, for controlling diseases, for controlling insects are all available under conditions when water is available and CO2 plus H2O equals CH2 and is working just fine. Right? Okay. So it would not have worked out without this component. Okay. So I'm telling there's a lot of geneticists in here, including myself. It's not just about genetics. Okay. That's what I'm trying to point out in here. Okay. So now the challenges of food security in the 21st century is illustrated in here. Okay. Let me just get you into the very gist of this one. Okay. So this is representing the projected population by the middle of this century, 21st century, about 10 billion people or more, right? Okay. So this graph shows the comparison of the current yield using the Green Revolution technology of some of the major crops, maize, rice, wheat, and soybean, okay? In order to feed this population increase in here, okay, we need this increase in genetic potential to sustain the yield that is necessary to feed all of these people. Under conditions when much of that environmental factors that makes the Green Revolution paradigm work is no longer here. Okay? And therefore, we get into the second challenge to food security in the 21st century, scenario number 2, the climate and the ecology question, which is a lot of us in here are probably missing. Okay? So will there be enough natural resources to sustain agriculture? Unfortunately, as successful as Green Revolution was, there was an unintended consequence. Number one, the erosion of soil health, because it was overutilization of natural resource, depletion of water, pollution of water, right? And therefore, agriculture, as ideal as it was during that generation, it's no longer ideal now, okay? And put another big circumstance in there, put climate change in there, right? Huge, okay? We cannot think in a very, very simple way by thinking that we can find a gene that would make a rice plant grow and produce well under condition when water is limited and where everything is polluted, okay? So, moonshot challenge to science, okay? I quote, JFK. So he said, send a man to explore the moon, right? So I quote, we chose to go to the moon in this decade and to do other things, not because they are easy, but because they are hard. Because that goal will serve to organize and measure the best of our energies and skills, because that challenge is one that we are willing to accept. Okay, one one that we are not unwilling to postpone, and one which we intend to win. Okay, so look at that context in what you all are doing now. Okay, as plant scientists. Okay, the moonshot challenge to agriculture now. Okay. Anybody else seen this movie? Right? Okay, right. So what is the missing there? Okay, all the ideal components of the Green Revolution paradigm. So is it a moonshot challenge? It is. It is a moonshot challenge. It's as big as finding a solution to cancer, in my view. Okay. So another problem here. Okay, because of this paradigm that I showed you a while ago. Okay, this is what we are working with right now. Okay, this is the diminishing extent of diversity, genetic diversity from the natural population back during the time of hunting and gathering, okay, to the time when humans selected few of those components of biodiversity to be grown and cultivated under well-managed conditions, which we call agriculture, okay? So the diversity is very, very low now, okay? We're talking about how crops can adapt to the changes in the environment. It's probably not here because these were selected under condition when everything is ideal. Right? So where are the mechanisms that we are looking for to create drought-tolerant plants? Probably not here, probably not here, probably here. Okay? But the problem is that this is so damn difficult in terms of bringing them back to the genetics of the modern day cultivars. Okay? Fast forward again, milestone leading to the promise of the genomics revolution. I wanted to share this with you because this is the main point of what I'm talking about today. Whether genomics is enough, whether biotechnology is enough, okay? So from Mendelian genetics, okay, we move out to all of these Nobel-winning discoveries that led to what we call now genomics technology, okay? Right now, what you guys are working on is the Solexa Illumina technology that allows us to sequence every single individual in the population that we are working on for a tiny fraction of what it used to be back in the beginning, okay? So therefore, you can sequence, one genotype for a fraction of, hundreds of dollars, probably even less, okay, depending on what platform that you use, okay? So the technology is there, okay? Very, very impressive, high resolution technology, okay? Now, the new millennium, Y2K, okay, anchored the genomics revolution. Why I'm showing this to you? Because I wanted to show you that technological revolution also led to this important revolution that we call genomics, okay? At the dawn or the turn of the 21st century, you can see the progression of how many genomes in animals and plants have been sequenced, right? Okay, and to use the human genomics as an analogy, okay, I would like to pose this challenge to most of the students in here. The biggest promise of genomics revolution in human biology, did the human genome revealed all the answers, okay? When the human genome, the first draft of the human genome was released in the journal Nature in 2001, this was a big promise. We will identify all the genes that define the blueprint of human biology. Cure for cancer, even aging, all of those kinds of things. If you read, popular literature, one of the biggest topic in human health now is what? Aging, right? All of those neurological diseases, okay, that goes with aging, okay, that's part of the promise of human biology, okay. But as we see the progression of information and technology, okay, they keep changing the paradigm. Here we will identify everything using the genome, okay, and then later on they found out that, well, the genome doesn't act alone, okay, because the microbiome, all of the microbes that co-inhabit our body is responsible for how this genome works, okay. And later on, they found out that knowing the sequence alone doesn't really allow us to understand biology because there's a second language that is hidden that we call now the epigenome, right? So on and on and on and on, from a very big reductionist promise to something, the realization that, it's not as simple as that. Okay. So what's the lesson that I learned as an outside plant biologist? Biology and how organisms function in the dynamic environment they live are too complex to be explained by a reductionist perspective. Okay. So the big promise of functional genomics, okay, this is what the genomics revolution was saying, okay, we will understand all the functions of every gene, okay, and use that, you know, to cure diseases, and in agriculture to create new genetics that would produce more food for the future. So this is the holy grail, right? So, but let me quote on this. Despite decades of research since the first sequencing of an organism in 1995, scientists still do not understand exactly how the information in genes is converted into an organism's phenotype. Why? The biggest thing that is missing in this paradigm is ecology, environment, right? Because genes don't act alone, okay? Ecology is necessary. So could functional genomics do it alone? Okay, so let me walk the students through the paradigm of functional genomics, how it's done. Okay, you have a model system. In this particular case, I'm showing Arabidopsis, and there are several plant models as well, not just Arabidopsis. Rice is also considered to be a model. Okay, so we do this forward and reverse genetics. Okay, forward genetics is that you identify a knowing the phenotype. Okay, what's the causal gene behind the phenotype, okay? Reverse genetics is that, well, we don't know what this piece of DNA in the genome is doing, therefore we introduce mutation into it, okay? And if it leads to a change in phenotype, then we know the function of that gene, right? Okay, simple. For this is not new. This was even during the Drosophila genetic era. People just made it appear like this is brand new. It's not, okay? And then whatever we learn from gene functions here, we apply that to crops, okay? Given the assumption of orthology, paralogy, and xenology, and all those kinds of things, synteny, right? Okay. But what we underestimated is the fact that adaptive traits cannot just be a single gene, just like what the Green Revolution one did, right? Okay. So adaptive traits, again, is a conglomeration of so many different perturbations, okay, that all feed into making that CO2 plus H2O equals CH2N inefficient or term, right? So we cannot address that with functional genomics alone. Could transgenic actually do it alone? Okay, so I'm a molecular biologist, so I was trained initially, okay, using this paradigm of transgenic approach, and I can tell you, back in the late 80s and early 1990s, we were some of the pioneer people who were actually starting to utilize this as a component of the breeding toolbox in rice, okay? But let's look at this within the context of adaptive traits. Many genes, and I will show you how complex it is in a while ago. Many genes, okay? But with the transgenic approach, you can only manipulate or alter the function of one gene at a time, okay? So let me give you an example here. The golden rice, people have heard about this, right? What was done in the golden rice, okay? Rice lacks beta carotene, okay, in the endosperm, okay? Beta carotene is required to enhance the nutritional value of rice, particularly in regions in South Asia where childhood blindness was very prevalent because of the lack of the vitamin coming from beta carotene, okay? So the pathway that leads to the production of beta carotene in rice is there, but it's not functioning because some steps in the pathway, as I have shown with crosses in there, is not working because the allele of the gene that encodes for those enzymes is a mutant and therefore non-functional, okay? So people use this as a model, the daffodil, and they identified a functional allele for those genes, okay, because it produces yellow color and essentially creates single transgenic line that expresses the correct allele for each of these steps that is missing in rice, okay, and make those to express only in the grain, not in the leaves, not in the roots, only in the grain, right? So single transgenic lines were then hybridized and bred to have all of those correct alleles, and the result of that is a lycopene and beta carotene producing rice, right? Easier to do now in today's standard, right? Why? Because you are just adding the correct function in a pathway that exists, okay, that leads to the production of just one product, carotene, right? When we're talking about adaptive traits, it's not just one product. Probably thousands of products, and the interactions, the permutation by which all of those products interact, okay, is enormous, okay? So could gene editing do it alone? Okay, this is one of the most significant, in my view, achievements in CRISPR-Cas system in plants in the recent time. Okay, so what this study has done is essentially to go back to the progenitor of the modern day cultivated tomato, okay, and test the hypothesis whether can we make this progenitor of the cultivated or domesticated tomato, okay, mimic the phenotype of the domesticated tomato that has been domesticated by humans for thousands of years, right? So by gene editing, correcting or converting, mimicking the alleles for these traits, okay, in the genetic background of the wild, okay, they succeeded, right? They were able to convert the phenotype by altering the allele, right? By gene editing, okay? This is great, right? But the question is that domestication traits are not are Mendelian genes, but resilience and yield plus domestication is so much more than altering the function of 4, 5, 6 genes, right? It's probably an entire genome, manipulating the entire genome. Why I think we may be missing something? I think there is an oversimplification of the complexity of ecophysiology with a gene-centric paradigm. What do I mean by that? Okay, so this is one where Jen and I have talked about this book a number of times, The Music of Life. If you guys have not read this, read this. Okay, it will enlighten you. Okay, very short book by a very prominent human physiologist, Dennis Noble. So to summarize this, what his theory is, okay, physiology is so complex, cannot be explained by a gene-centric approach alone. I'm not saying that genes doesn't have anything to do with it. Of course, gene has a lot of things to do, but if we reduce the complexity by just looking at one gene at a time, okay, without considering the interaction and how the environment affects that interaction, we're gonna fail, okay? So, what it means is that physiology is a consequence of the function of genes and the network of genes. Once physiology is created, physiology feeds back into the functions of genes and alter the activity and interactions of those genes, okay? And to add more complication, let's put the environment there, right? So it's really very, very complex. So to quote this, okay, a student, I believe, Kate from the University College, Oxford, reacting to this book, okay, in the context of reductionism versus holism. The book's alternate title, Biology Beyond Genes, explains Noble's main aim to show that organisms are more than machines built by genes. He demonstrates that the relationship between organs, tissues, cells, organelles, and genes isn't always bottom up, okay? And that these higher systems can affect gene expression. In school, okay, we are often taught that there is a gene X, okay, a gene for X and a gene for Y, right? Okay, Noble combats this by explaining in simple terms that multiple genes and environmental factors need to come together in order to produce a specific phenotype or function, particularly within the context of adaptive traits, okay? If you're talking about, okay, white color, petal color versus red petal color, that's essentially Mendelian, okay? We're talking about how the system, how physiology interacts with the dynamic changes in the environment, okay? So I think there's an oversimplification that we could create, you know, the next generation of drought-tolerant crops by just doing simple genetics. In my view, it can't be done, okay? So here's another thing, why I think we may be missing something. Cellular energy allocations is equal to the penalty from perturbed source and sink dynamics. What do I mean by that? Let's go back to this most important chemical reaction in the universe, okay? Let's perturb this. Let's remove this water, this component, right? So water is needed, right? Photosynthesis is not going to work without water, right? So let's look at a situation wherein this is available, right? So environment is optimal. Say the CH2N is the product, okay? It fuels everything that happens in an organism. So therefore, in terms of cellular energetics, this, I believe, is the logical partitioning of the products of photosynthesis into these categories, okay? One sink is for growth, okay? Development and productivity. This is the purpose of the plant, to reproduce. right? And give rise to the next generation. Of course, plant will have to adapt to changes in the environment, and therefore it needs defense and maintenance, right? In this particular case, because the environment is optimal, just like in the Green Revolution, a smaller proportion of this sink is defined by maintenance, right? So let's look at when we perturb this and remove this or reduce the water, right? So let's look at the distribution there between sink and source, okay? So you see the alteration of the distribution, okay? Much of that source is getting into defense. Therefore, less photosynthate is going to get into reproductive tissues, which we call yield, right? Simple. It's not free. Right? There is a penalty for that, okay, as a consequence of the change in environment. Another reason why I think we may be missing something, so this is a new theory or a new paradigm that was proposed by the human genetics community, okay? This was proposed in the 2018, 2017, like that, okay? So this is what they call the omnigenic theory, okay? In classical genetics, we define some traits as polygenic traits, okay? Therefore, there could be tens, dozens, and hundreds of genes that contribute in a cumulative fashion to a complex trait, which we call quantitative trait, okay? But the way the human biology community looked at this is in context of the theory of omnigenic traits, and they propose that the entire genome is involved, right? And how do they put this forward? They propose that there are two components of this, the core or cis effect genes, which are shown here by green or yellow, depending on who's looking at it, okay? These are the major genes whose effects, okay, is quantifiable. Effects on the phenotype is quantifiable. Therefore, if you mutate this gene deliberately using functional genomics, you're gonna see a reduction, okay, in the phenotype, okay? Okay, so they have defined effects on that. But what this theory was proposing, there are many different components of it that are called the peripheral or trans-effect genes, whose individual effects cannot be quantified. But if you combine the cumulative effects of all of that, their effect is probably greater than the effect of these core genes, okay? But in order for them to work, the right alleles for the core effect and the peripheral genes all need to come together. Just like what Noel was saying, right? If you mess up some of these alleles, you're going to have a reduction, right? Very complex kind of interaction there, okay? So another thing why I think we may be missing something, I think the diversity used in functional genomics does not capture the needed genetic novelties. What do I mean by that? I have explained this in the previous slides, okay? Because of the process of domestication and targeted selection in modern day plant breeding, the amount of genetic diversity from the original population in nature has substantially got reduced and explained by this genetic bottleneck effect. So the diversity that we are looking at these days is just this. So perhaps many of those traits that were not incorporated during the selection, domestication, shown in this smallest bottle in here, are probably not here, are probably here. We need to go back, okay? But the problem is that, again, it's difficult. It's not easy to do that, okay? I've had a lot of heated debates, with plant breeders in the cotton community that, challenging my dogma that, why are you repolluting the modern day cultivar, the germplasm of modern day cultivars? We just have to find a way to further enhance this to adjust to the current conditions that we call agroecology. How should we refine our research paradigm? Again, I'm a very big supporter of the importance of biodiversity, okay? So let me walk you through this specific example here. This is a data that I got from the International Rice Germplasm Research Center at Ely, okay? So this summarizes the total amount of genetic diversity that is being held in the germplasm bank, okay? 132,000 accessions, okay? The breakdown of all of these components are shown in this pie chart in here, okay? So the traditional cultivar landraces, that means not subjected to breeding, okay, other exotics that they don't even know what they are, okay, in terms of genetics, okay, you can see the proportion of these two classes in the entire holdings of the germplasm bank, okay. Of course, there are wild species in there, and there are some weedy cultivars in there as well, right? So now, this is the distribution of how much of this diversity, okay, is actually coming from the different countries that actually grows rice, okay? I am highlighting India, Indonesia, and Philippines because that's my main interest. That's a germplasm that I currently look at as far as my research is concerned, okay? So the bottom line is that only a very small portion, less than 5% of these 132,000 accessions were used to bred the modern, the Green Revolution type of genetics, which we are continuing in today's time, okay? Biotechnology and genomics work on a very small proportion of this total diversity, which is only captured by this, okay? So how can we bring in new traits that is relevant to the change environmental condition if we are just focusing on that very limited amount of genetic diversity, okay? So again, for biodiversity, giving a specific example in here, we're looking at this Pokali, which is an Aus. This is the diversity panel of rice, okay? Aus belongs to group 2, which is shown in here. For those of you who are from India and from Bangladesh, you are very familiar with this in the Kerala region. This has been grown and adapted to condition where irrigation water is brackish water, right? And they survive. Yes, they are not as high yielding as modern day genetics type of cultivars, but they produce under that suboptimal environmental condition. That's one. I'm also currently pursuing this because I'm from the Philippines. I'm also pursuing some important things that I have observed in the Philippines. 500 years of cultivation in the highlands, no irrigation, relying on rainfall, right? And this was cultivated by the Ifugao tribe that has been in existence for the last 1,000 years. Right? And they were not colonized subject to the colonization of Spain, of the country, and therefore the culture is very intact, agriculture is very intact. So I am pursuing this because I believe there is something hidden in it, right? Because I believe they represent, you know, a micro environment and a certain type of genetics that co-evolve together. It's just a matter of us identifying how those works and asking ourselves, can we bring it to modern cultivar? I know it's not easy, but we have to start asking those questions as scientists. Okay, again, for biodiversity, wild gene pool in rice breeding and biotechnology, the diversity is available, right? This is the representing 15 million years of speciation in the genus Oryza and 5,000 years of domestication. Okay? Dr. Shim is sitting in the audience. She has worked a lot on this particular topic. Okay, so a lot of these genetics can actually be brought into the genetics of that narrowest bottleneck and test the hypothesis: can we create new genetics? Can we create new networks? Can we create phenotypes that would mimic, okay, how plants behave when humans were just doing hunting and gathering, right? The problem, again, is just, it's hardcore science. It's rocket science. It's difficult to do it, okay? Dr. Shin knows that, okay? Because, you know, imagine two species that have been separated by millions of years evolution. You're suddenly forcing them to mate and produce an offspring, okay? You need hardcore science, but I think the technology, genomics and biotechnology, is right to be able to start addressing those difficult questions, just like JFK said, right? Addressing the difficult question. Don't back out on it, okay? Face it, okay? So understanding domestication in context of today's challenge to create novel genetics for marginal environments. So what the message I'm trying to share with you today is that this is the pathway that led to the domestication of plants that created the basic scaffolding of what we use in agriculture, okay? This is still ancient, okay? The domesticated version is still very ancient because we humans further improved them, okay, through genetics, okay, through breeding, of course, through management as well, okay? So you can see here the parallelism of domestication process in dicot group of plants and monocot group of plants pretty much eliminated certain types of traits, okay? Let me give you an example here, shattering, seed shattering, okay? So why is that an important component of domestication? If your crop shatters, what's the point in doing all the management where you're gonna lose the yield, the grain, because the grain shatters before you actually are able to harvest them, right? So that's one of the important genes that were fixed during the process of domestication. But the downside of that, perhaps, in my view, unknowingly, during the elimination of those other traits, there are other adaptive traits that unknowingly was being eliminated as well. And therefore, no longer present in this smallest bottleneck that I am showing you here. So again, okay, maybe we need to bring back some of the traits or alleles that were left behind during the process of domestication. Yes, it will pollute, okay, this germplasm and probably create some negative consequence, but that's why we have genomics. That's why we have biotechnology to fix those problems, right? Okay. So what is a promising approach to create the needed genetic novelty? I was in the Plant Genome Conference in January. So, my lab is with me in that conference. I think some of the other professors here, Dr. Patil was there as well. I was so inspired by one of the last plenary speakers from the University of Illinois, Urbana-Champaign, who talked about crop modeling. And therefore, Glenn, this is an inspiration that I shared with you for potential new avenues the department should pursue. So what is this, okay? So essentially, this paradigm is essentially capitalizing on the omics technology, information generated by omics paradigm, and translate that into AI, artificial intelligence, right? We are all talking about AI now. How does it work? Using all of this information that are unconnected to each other, artificial intelligence by virtue of algorithms try to see connectivities, okay? And then relate that to what we already know, okay? For example, we know how photosynthesis works, okay? AI actually aims to see if you can perturb this level of complexity, let's say transcription, okay, of certain genes, or translation, or protein maturation, okay? And see how that affects leaf morphology or anatomy, how it creates a new behavior of the root, okay? And how does it translate into plant productivity, okay? Very, very, very impressive, okay? This is, a huge multidisciplinary program at the University of Illinois at Urbana-Champaign. So what I'm proposing in here, I'm not proposing to abandon the functional genomics paradigm. It's very, very powerful, okay? We all do it, okay? But let's put it in a different context. The purpose of functional genomics paradigm is essentially to identify what genes does, right, in the cell. Doesn't mean we know what phenotype it creates, okay? In the context of agriculture, at least, okay? So that's a function of that. So we understand gene functions, molecular, biochemical, growth, developmental, and all of this, right? So with that knowledge, okay, we should use that to infer what traits, according to the results of functional genomics, matter to create a new immediate type of plant. Just like what Dr. Cruz and Norman Borlaug did during the first Green Revolution, they identified, plant height is important, right? Flowering time is important, right? Elongation, internode elongation is important, right? Let's identify those individual traits, and that's how we all should be able to contribute, okay? No one here can claim that you hold the solution because you have the most powerful technology at your hand. All of what we do is needed in order to address that particular challenge. So still idiotype, okay? But let's enhance our ability to identify what are the traits that are necessary in the new idiotype that would fit very well under conditions when water is not very abundant, soil health is very poor, nutrients are not available, climate change is creating all of these, ridiculous things that, make California suddenly have tons of water when in fact California is supposed to be experiencing drought. Okay? So look at that in the big picture. So imagine how the outcome of this multi-scale plant modeling that utilizes information from the genome to phenome to biochemical pathways to physiology, okay, tell us about what traits and what physiology, what anatomy is important to make a plant survive and produce grains under conditions that we call drought, right? So this is powerful in my view if we use it correctly, okay? This is my vision on how we could actually move forward, a realistic approach. Of course, for all of you, your goal is to get your PhD done and publish a paper, right? Okay. But I hope with this, you're enlightened such that when you write your first research proposal, okay, as an assistant professor, after you complete your PhD, you're not going to promise that, oh, I'm going to create a drought tolerant plant because I have, one transcription factor that I'm going to overexpress. That's BS in my view. Okay? I'm being very candid in here. That's BS. Okay. And again, I don't expect everybody to agree. Okay. But my goal in here is to inspire debate, to inspire critical thinking. Okay. Because if we don't do that, we are all going to continue, focusing on what we do in our lab without really putting what we do into the context of the big question, which is the moonshot challenge to agriculture in the 21st century. More lessons from the big scheme of biology in favor of holism. Okay? Summarizing that, evolution optimizes by trial and error, leading to the most optimal interactions at multiple levels of biological complexity. Okay? Therefore, this is an effect creating the next generation of climate resilient or environment resilient crops. Okay? It's kind of like mimicking the effects similar to natural selection, survival of the fittest. Always remember that, okay? Don't remove the Darwinian principle in what you all do, regardless of what level of biological complexity you are addressing. It is important. Believe me, if you understand this, you're going to be an excellent biologist because everything is based upon this particular theory, okay? So this cannot be hastened by a one-shot genetic approach, okay? Even green revolution is not a one-shot approach, okay? Because there are multiple genes that have been stuck in there. So let me give you an recent example of the complexity of evolution, right? So there was a big BMGF program based in the Philippines at IRI that aims to convert C3 photosynthesis to C4 photosynthesis. Why people want C4 photosynthesis? It's a better performing type of photosynthesis under conditions of low moisture, high temperature, low nutrients. Essentially what Green Revolution 2 is all about, right? Okay. So, but unfortunately, rice is C3, which is an older or ancestral type of photosynthesis. So this was separated by 30 million years of evolutionary history. From C3 to C4, it was created by co-evolution of many traits, enzymology, biochemistry, anatomy, development, okay, in order to create the more environmentally resilient form, which is C4, right? So the paradigm is that, okay, let's use genetics and convert rice, which is C3 to C4, okay? The challenge is like this. We need to change morphology, anatomy, enzymology, biochemistry. It's not just about, okay, let's replace Rubisco with PEP carboxylase and everything's going to work out. There's a lot of anatomical aspects to it because, again, one of the critical aspects of C4 photosynthesis is cell-to-cell exchange. Once CO2 gets inside the leaf, right? So this is engineered by Kranz mesophyll cells, the Kranz anatomy, okay, which involves a lot of genes. So it's not just one pathway you're going to have to stack together. It's very, very complex. But do I think it's doable? Probably it is if we to create something new, a new crop, okay? But to convert rice to from C3 to C4, I don't think it is really, I think it's bordering a flawed kind of thinking in my view, okay? So I'm gonna close by showing you this. This is the model that I've created several years ago, okay? The apple tree paradigm or model, okay? So here you're seeing the apple tree here that's full of fruits, right? Okay, the totality of the fruits in this particular tree is essentially the genetic potential of that particular tree, whatever it can produce given its genetics, right? So this is right now, the maximum is the level of productivity, the level of yield we need in order to combat the grand challenge, the moonshot challenge to agriculture, okay? This is what we need in the future. So let's look at green revolution, okay? In my view, green revolution paradigm was relatively simpler, less complex compared to the genetic complexity that we need to do now in today's time, okay? So the ladder in here represents the technology, okay? So it picks the low-hanging fruits in there. That's enough, okay, during that period because, again, the population is not as big, right? The 50 billion, the 10 billion, 10 BPQ, the 10 billion people question, right? Okay, so let's move forward. What did the genomics revolution, the recombinant DNA era did? Transgenic marker-assisted selection, functional genomics in the 80s and 2000, the gene-centric paradigm. It creates a lot of refinements that allow us to maintain, if not increase that yield a little bit, just a little bit, okay? But it's a powerful technology that allow us to continue on the success of green revolution, okay? But now the realization is that we need to pick the highest hanging fruits in there. It's too high, so we need the highest ladder that we can afford. That using the reductionist approach, which is in this ladder and in this ladder, is not going to work anymore, right? We need to face the difficult questions and the difficult integration of modern technology in order to create this highest ladder that could bring us to the highest complexity of biology that could bring us those additional yields under conditions when environment is becoming more and more suboptimal or around. I was hoping that there will be some people from other departments in the social sciences that would be interested to attend this talk, and therefore I included this. This is not just about genetics. Let us not forget about the socio-political economic aspects of food production and how food produced by agriculture is distributed. Okay, let's not also forget about the socio-political economic aspects of natural resource utilization, particularly water. Okay, I'll share you one first-hand experience. Many years ago, I think 2017, I traveled to Punjab. Okay, you know, which is like the bread basket of the world. Even now, the yield of wheat is unsurpassed, okay? And not only that, I have observed with my two eyes, the vegetable production is just enormous, okay? The yield potential is very high, okay? But then I see that they're just being disposed, right? As trash, like really enormous broccoli and cauliflowers are getting disposed. So I asked my mentor, Dr. Brar, Darshan Brar, Dr. Brar, why are those harvest? They're very good quality. Why are they being discarded? Because there is no means, efficient means to transport them in different parts of the country where they are most needed, right? So genetics and agronomy and physiology and crop protection and soil fertility are not the only components of this equation. There's a lot of it that is associated with social, political, economic aspects as well. Okay, so I hope I was able to get your brain thinking and get you inspired to ask bigger questions. Continue what you're doing now, okay? Continue the excellent technological breakthroughs that you're aiming to do as students of science, but don't forget the big question. Don't forget the philosophy, and certainly don't forget the history, because we should learn something from the failures and successes of the scientists that, you know, did it even before us, right? So with that, you know, I'm just showing you this. This is essentially what makes what I do at Texas Tech possible. Okay, these are some of my key collaborators that are allowing me to pursue these big questions within the context of what society really needs. Okay? My advantage here is that I'm a full professor. You know, I don't need to get promoted. I can pursue whatever I want. As long as I can convince the funding agency that my science is worth funding. right? That's my, this is the most enjoyable part of my career because I'm able to pursue those big questions without having to worry, wait a minute, I have to get promoted to associate professor, therefore I cannot do this. I have to focus doing this, right? So that's my my stage right now. And of course, you know, NSF and USDA, NIFA, and the previous iteration of USDA NIFA has been very, very much instrumental in allowing me to deliver both the successes and failures that I have done in my career. So that's my message to all of you today, okay? Think outside the box, okay? Don't forget about philosophy because your degree is going to be what? Doctor of Philosophy, right? So philosophy is very important. Okay, I'll be happy to entertain some questions if we have time. If not, then we can have discussion. It's complex. We are the resolution by which we investigate genomes, okay, or all levels of biological complexity is incredible these days. So we shouldn't be studying only the cultivars or the modern genetics just because that's what the industry wants us to study, right? We also need to invest our resources, genomics and biotechnology, to understand how those natural components of the gene pool actually works in their native habitat. Okay? We need to learn from them. Okay? As far as technology is concerned, I mean, you can sequence anything these days. You can do transcriptome analysis on anything. Okay? But the other thing as well is that when I was doing my PhD, everything that I do, the naivete of me when I was young, is that, okay, I do the experiment in the greenhouse or in the growth chamber. I always think that this answers or solves the problem. Okay. Now we need to go back to the actual ecological condition, right? And therefore, that's why I go to Ely, because, you know, we do our field screening in a coastal area, and that's where we plant our rice. Okay, so when we manipulate certain genes or certain loci in the genome, and then we test that not in the greenhouse, not in the growth chamber, but in the actual field that farmers use to grow rice that is constantly or perennially affected by salinity stress condition or drought stress condition. So to answer your question, the technology is there, but we just have to invest on fundamental research that allow us to learn the intricacies of molecular, biochemical, physiological mechanisms that makes those components of the natural diversity fix best under natural ecological conditions. And again, in the abstract that I circulated, I said the technological advance is right. Okay, we just have to use it properly, okay, in the right context.
Any more questions? Yeah.
There's a question there.
Yeah, thank you for informative presentation. Just I have one question. How artificial intelligence can bridge the gap between Gen 5 and Gen 5?
We have been doing artificial intelligence. For those of you who might think that this is new, it's not. We have been doing artificial intelligence for many, many decades now. Okay, let me get to the example of that. Algorithms, right? So what are algorithms? You supply raw information and let the algorithms connect those dots, okay? And then create a conclusion that may have some relevance to the biological process that we are looking at, right? So we have been doing that, except that it's not as efficient as we think it's going to be efficient in today's time, okay? Number one, because today we have so much more advanced computing technology, right? Okay, we can handle large amount of data sets, okay, and integrate that. Okay? During my time, it's just relational database. For those of you who are doing genetics, you know, during my time, if you clone a gene, sequence it, a DNA sequence it, and then you blast it against NCBI, non-redundant databases, there is probably 50% chance that if you are working on an orphan organism, that a homologous sequence is not yet in the database. And therefore, the artificial intelligence, which is BLAST, okay, is not going to be able to tell you what it is, right? But in today's time, science and governments have invested so much on all of these different layers of biology. It's just a matter of using this AI approach to actually create those brains, the neurons of biological brain, okay, to integrate all of this. So going back to your question, in Illinois, what they're doing, okay? Let's say you have all of these layers of biological data, okay, from transcriptome to metabolome to genome, transcriptome, metabolome, biochemical pathways, and phenotype. What are the ranges of phenotypic changes that occurs in plant across in space and time, okay, spatially and temporally, okay? By integrating all of those data, this artificial intelligence would then be able to ask the question, let's perturb this. What does the perturbation in this level, how does it impact the next level and the next level? And how does the root behave? Is the root affected? Okay. Is the leaf growth and anatomy affected? And therefore, you're able to test what traits matters, okay, in space and time, right? So this is, you know, I wanted to reiterate this because I think as a department, we have the expertise that spans the spectrum of what those omics AI or crop modeling is all about. Okay, we just have to think and discuss how we can create new ideas. Okay, because I think the expertise is here. It may not be as a huge of an expertise as other universities, but certainly I think all of the expertise leading to that paradigm, I think we have it in the department or maybe in the department. Okay.
Yeah, fantastic talk. I appreciate you making it accessible to a non-geneticist especially, but I'm curious about the soil side of this. Specifically, you were talking about the environmental interactions with genetics, and I'm curious, what do you think is the lowest hanging fruit that the manipulation of genetics, genetic engineering accomplish that would change the soil properties? Is there a particular soil property, or maybe I'm thinking root architecture or something that would make specifically for crops around here, like in cotton, more?
This is how I view it. Yeah. So when we look at the interaction between a pathogen and a host plant, okay, I always tell this to my students, okay, you cannot just look at either the host, okay, or the pathogen genetics, okay, because these two interact with each other. Going back to the soil, your rhizosphere, the root, okay, I think it would be worthwhile to actually look at the diversity of physical attributes of soil, okay, and compare that with the nutrient contents, okay, across these different, you know, physical attributes of soil, back when the environment is not as limiting as it is now, okay. But then again, you cannot leave from the equation the fact that for you to understand how the physical changes and chemical changes and microbial changes in the soil happens, in order to understand the biological consequence of that or implication of that, you need to understand how plants react to it. What type of genetics of plants, you know, react to it, okay? And what is the outcome of that interaction? So again, we cannot just work alone these days. We just have to do a cross-disciplinary. We need to talk to each other and actually facilitate this novel interactions or collaboration that hasn't been done in the past. That's my thoughts. Yes, there's some.
I have a question.
Yes.
You told us like, okay, we have high yielding plants, rice, but it depends on the sun, environment, every factors. It was great in green era, and now the changing environment, urbanization, everything. That crop, the variety may not be as yielding or surviving as it is now. So my question is, this is going to be changing all the time. Like environment, like, yeah, it is moving. And we don't know whether it's better or worse. But my question is like in the same time, some wild type variety, not manipulated, not given, they're also evolving. Are there any groups that you know studying them, like today's wild type, and can actually study.
What is most affected, okay, is not really, yes, I understand, okay, there's a natural evolution in all different types of environments. But I think compared to the magnitude of impact, okay, the effect on cultivars that we grow under very well managed artificial environment that we call agroecology is so much more prone, okay, to all of these changes, right? Okay, and therefore the penalty is going to be very high. So Darwinian principle, okay, survival of the fittest, natural selection, okay. So relate that to the concept of extinction, for example. Yes, I know that the climate is changing, environment is changing. Of course, all of these wild species that are in nature are also affected by this, right? Okay. But it will take, you know, an evolutionary time in order for extinction doesn't happen, okay, in a period of 100 years,
Yeah.
right? Extinction happens over a period of long durations of time, right? And therefore, because that's what we call them wild, exotic, they can pretty much handle all of these changes in the environment. And another thing that I wanted to interject in that, okay? So if a wild species is affected by drastic environmental condition, okay, its ability to produce seeds, okay, is impacted. But does it need to produce, you know, certain tons of seeds per hectare?
Yes.
It does not. As long as it produces some seeds, the next generation is ensured, right? So we have to think in that kind of paradigm, right? Agriculture is artificial, okay? We cannot just say that, okay, we're going to be generating all of this climate resilient type of genetics, okay? Just like how it occurs in the natural environment, when actually the diversity that we are looking at is very, very narrow. Yes,
Go back to
I
AI.
didn't expect I'm gonna get this.
It was thought provoking, so you have this.
What type of data AI analyzes? More like text or images or?
All,
Oh, so you guys have too much data.
all layers of biological complexity. Genome, transcriptome, metabolome, phenome. So when we say phenome, it's so many different levels of complexity, not just anatomy, it could be ultrastructure, you know, all of those levels of complexity. And you'll be surprised because of the open data sharing policy of, you know, federally funded programs. These data are available, okay, in the public domain, right? So some of you might be thinking it's very hard. Maybe I should switch to more computational aspect of biology, because I believe that this is really an important power that would allow us to learn something new. Okay, I'm not saying that we're going to have a solution, but I think we're going to have something new that we can test and see if that's going to be a tangible solution that agriculture can activate.
Is there any particular side goes on the data? Or is there any particular side?
Unfortunately, they're everywhere.
I just have to.
You know, of course, the simplest solution is NCBI, National Center for Biotechnology Information, that holds all of those different types of data sets, okay, including the description of all the experiments on how those data were created, okay, or were generated. Okay, it's not perfect, okay, but I think that the volume of information that's available in the public domain really justifies that we really need to test this AI kind of an approach to model a plan. I'm not saying that, you know, the digitally modeled architecture and digitized and physiology of the plant is what farmers should be planting. This allows us to test those models in actual field, right? Imagine the ability to create different phenotypes, okay, without really investing so much first on breeding them, okay, but testing them first, okay, artificially, okay, by artificial intelligence. And the best possible outcome of that, let's breed this and test it in the field. That's simplistic and that's how I see it. So of course, I'm not an expert in AI, but again, I'm trying to see if there will be some few brains in here that could be inspired to integrate that into what you guys do.
Yes, sorry to interrupt. We are running out of time.
Yes, yes, okay. Yeah,
Thank-
thank you so much for listening.
Thank you everyone for making this time to make this happen. So keep an eye on your email. So each week we have a seminar like
Yes,
this.
yes.
Maybe this room not be available, so we'll send you an email with update. So keep your eye on the email. Thank you so much.